Hi, everyone.
Hello.
Good afternoon.
Hello.
Hello.
How's everybody doing today?
Good.
How are you, Professor?
I'm
Good.
okay. I know everybody's getting tired, right? We're nearly close to the midpoint of the semester, so fatigue starts to set in. So hang in there. I know it's not easy. Okay. So welcome back, everyone. I hope that you and your families remain safe in terms of COVID and things like that. Tiffany, thank you for being here. Okay. So let me just start off with some business items for today. Actually, yesterday, Elena met with me and she informed me of a typo on the lab activity sheet. And I simply wanted to comment about that. So let me bring this up here. So in the lab homework that you have due for Thursday, okay, the origins of antibiotic resistance homework, please be aware that there is a typo in one of the questions, question letter M, okay, question letter M. In question M, I had referenced questions G and H. That would be incorrect. Okay, so the proper question is, why do you think bacteria evolved the strongest resistance to antibiotics that attack the structures you identified for questions K and L? K and L are the proper questions to be referred to in this question. Okay, so I apologize if that gave you a little stress, simply a typo. Please take note of that to make that correction in your document if you haven't already surmised that there was a typo. Okay, all right. So thank you, Elena, for bringing that up. And do you have any thoughts or questions about that? Okay, so looking at your syllabus, today hopefully will be our last day lecturing out of chapter 13. That's my hope. I think I can finish it. And then we'll be starting chapter 20 on Thursday. So if we look very carefully at the syllabus, next week is fall break, right? So that means the classes normally held on Mondays and Tuesdays will not be in session. Fall break, Monday and Tuesday. Next Tuesday for our normal lecture, we will not have class, okay? Because it's fall break. Everybody's supposed to take a little breather and just catch your breath and have a little bit of time off, okay? So next Tuesday we don't meet, but then the next Thursday we have lecture exam 2, okay? So here's what I'm going to do. I will release the study guide for lecture exam 2 this afternoon. I will send it to you this afternoon, and it is my hope that you can at least read it through once before Thursday's class, so that if you do have questions about what will be on the upcoming exam, we can talk about it this Thursday, because a week from this Thursday, you have lecture exam 2, okay? So I will send you the study guide this afternoon. Please review it so that if you have questions, we can talk about them this coming Thursday, okay? All right, any questions you might have for the benefit of everybody? Okay, folks, hang in there. And you know, I do encourage you to take a little bit of personal time next Monday or Tuesday to do something that sometimes you don't have the luxury of doing, even if it's simply sleeping in late, going out to eat, taking a walk, riding a bicycle, going to see a movie. You know, take a moment to do something for yourself, because when you're working hard, you gotta play hard too a little bit. Okay. Okay, so we are going to get back into chapter 13, but before we do that, we need to finish the review of this homework problem, right? So we've been at it now for, this is like the third attempt at this in terms of the protein, okay? So let me ask in the chat window, okay, for your homework, what protein sequence did you figure out from this activity? Okay. Please, everybody type in what he or she got, okay? All right, so I'm just giving you a moment to fill in your responses there. Okay. All right. Thank you, everybody that's contributing. Okay, I'm just digging out my notes from the other chapter here. Okay, for those of you that got aspartic acid, stearine, and valine, that is exactly correct. Okay, that is exactly correct. So this would be, the abbreviation would be ASP, stearine, and valine. Okay, that's right. So remember we said in one of the previous comments, remember we said this: DNA and RNA polymerases always make new DNA. They always make new nucleic acid 5' to 3', which means the enzyme has to sit on the 3' end of the template to do its work. Okay, so in the same vein as that, so that's something important to keep in your memory banks. DNA and RNA polymerases always make new DNA 5' to 3'. To do that, they have to sit down on the 3' end of the template to do their work. In a similar fashion, you should understand that ribosomes always sit down on the 5' end of the RNA template to begin translation. Okay, ribosomes always attach to the 5' end of the RNA template to begin translation. Okay, so that means when the ribosome began translating this red color-coded RNA on the screen here, it actually attached to the 5' end, and it came across from your viewing perspective right to left. Remember, don't think about right and left because the molecules and the machinery inside the cell don't care about that. Okay, so that means, to properly interpret this, I always recommend this. I see your question, kid. Hold on. I always recommend rewriting the RNA sequence so the 5' end is on your left. The reason I recommend you do that is because as human beings, again, we've been trained since we were children to read material across the printed page left to right. So we're comfortable with left to right, left to right. So to help yourself not make a mistake with this, take the RNA, identify the 5' end, and rewrite the RNA with the 5' end on the left, right? So that means that we would write the RNA this way, right? It would be GAC, GAU, UCC, GUA, 3', right? GAC, GAU, UCC, GUA. If I rewrote the top RNA sequence, if I rewrote it this way on the bottom, it's the same information, right? It's the same information. I simply put the 5' end that was on the right side on the top on the left side of the bottom. And then you use your genetic code table in your textbook to decode this 3 nucleotides at a time. So, okay, Kit, you're asking something here. Would Elena's question be the full, complete answer, or would the aspartic sharing valuing be acceptable if this were on the test? Okay. Kit, the honest answer is the exam question that will be related to this content will guide you to the proper format of how the answer should be. So Elena's answer is quite okay, but other answers are alternatively possible as well. The exam will guide you to the proper format. Okay, folks. So I cannot emphasize enough that the directionality of the molecules actually matters to the machinery that's manipulating the molecules. The directionality matters. Okay. Do you have any questions about this? Okay, all right, folks. So let me know. Again, on Thursday, we'll have an opportunity to talk about questions. All right, so for today, our talking points are going to be covering some basic ideas in bacteriophage's lambda life cycle and HIV. Now, we were talking last time about growing animal viruses. HIV is an animal viruses. We talked about embryonated eggs, cell culture, and living animals as possibilities for culturing viruses. Talked about some of the pros and cons of that. Now we're going to get into, well, what exactly happens when viruses infect cells, what goes on here, okay? So I'm going to talk about bacteriophage lambda. Bacteriophage lambda can be most appropriately thought of from figure 13.12, 13.12, figure 13.12. But I will also say, for the purposes of understanding the basic idea of what lambda is and what it does, that you can even study figure 13.11. Figure 13.11 is about T-even bacteriophage. But even though it is about T-even bacteriophage, there are enough similarities between T-even bacteriophage and bacteriophage lambda that it would be okay to study the material in figure 13.11 to understand bacteriophage lambda. So figure 13.11 is also relevant for lambda, even though the figure is technically about T-even phage. Okay. So I will also point out, if you're looking at either figure 13 or 11 or figure 13.12 in your textbook, you're going to see some artist drawings of what the bacteriophage looks like. I want you to know that those artist drawings of what the bacteriophage look like are actually pretty faithfully correct to the actual structure of the bacteriophage. And so let me turn your attention to figure 13.5. Figure 13.5 shows the structure of T-even, which is very similar to bacteriophage lambda. Okay, so if you have figure 13.5 available in front of you with your textbook, look at picture A. You see the bacteriophage lambda particle there. And in the upper right corner of figure 13.5A, you see a transmission electron microscope image. You see a scale bar there of 80 nanometers. And you see the actual viruses. Of course, the textbook has color coded them to justify making the textbook more expensive than it needs to be. But the actual structure of the virus is accurate. And so let me just flip up a quick image here of electron microscope images of lambda itself. Just take a look at some of these things, okay? For example, this one here, okay? This is an electron microscope image of bacteriophage lambda. You can see that we're not making this up. Scientists are not making this up. It's not like we're just drawing things out of our imagination. These little beasties really, really exist. And this is basically what their shape and structure is. Okay, so getting back to our talking points here, you can use figure 1312, 1311, and 13.5 as relevant information to understand bacteriophage lambda. So let me just start, in case it's not clear. Remember, when we say bacteriophage lambda, well, let me say this, okay? Bacteriophage lambda is a virus that specifically infects E. coli bacteria, okay? Okay, Elena, let me ask you a question. If I could ask you to unmute, please.
Yes.
Hi, Elena. Elena, as a human being, do you have to worry about your human cells getting infected with bacteriophage lambda?
I would say yes.
So, Elena, let me ask the question slightly differently, and then you can choose, you can stay with your answer, or you can change it. So, Elena, do I have to worry about an epithelial cell lining my mouth getting infected with lambda?
No, because it infects bacteria.
That's what I'm
It
glad.
affects bacteria.
That's right, Elena. Very good. So, Elena, stay with me. Okay, so I'm glad you changed your answer there, right? We do not have to worry about lambda infecting our human cells. That's correct. Good job, Elena. Elena, one more question. Do I need to worry about bacteriophage lambda infecting the E. coli that's in my intestinal tract?
Yes, because E. coli is a bacteria.
Good job. That's exactly correct. Thanks for participating, Elena. So that's my point, folks. We do not have to worry about these viruses making our own cells sick, but we might have to worry about these viruses infecting the microorganisms, which are in turn part of our body. And those microorganisms, if their behavior is altered because of an infection, might in fact make us sick or change the experience of life that we have. Okay. So, all right. So bacteriophage lambda is a virus that infects E. coli. Okay. Bacteriophage lambda, like itself and other similar bacteriophages, they typically have what I like to call a Jekyll and Hyde mentality. And so I don't know if you've ever seen any of the old horror movies of Dr. Jekyll and Mr. Hyde by, or that old novel written by Robert Louis Stevenson. Okay, it's one of my favorites. But at any rate, there is sort of a duality. There's sort of a caterpillar and butterfly duality of the bacteriophage lambda life cycle. Okay. Now, when I talk Jekyll and Hyde, here's what I'll say. Okay, so if you remember the old story of Dr. Jekyll was Dr. Jekyll. He was a crazy scientist, right? But generally speaking, he was an okay human being. Okay, so we have Dr. Jekyll here. Dr. Jekyll would be equivalent in the lambda life cycle to something known as the lysogeny or lysogenic portion of the lambda life cycle. And what I'll say here at this early time is that when lambda is in its Dr. Jekyll phase, when lambda is within its lysogenic phase, that the host cell that lambda is in this case E. coli, the host cell is not destroyed, it is not killed, it is not murdered, okay? So the host cell, E. coli, gets to live, okay? Staying with our analogy here, when Dr. Jekyll converted into Mr. Hyde, right, he was a fiend, he was a beast, he was a very questionable character, right? So when lambda is in its Mr. Hyde stage, that would be the lytic cascade or the lytic portion of the lambda life cycle. And the lytic portion of the lambda life cycle results in its host. Let's say you go to a dinner party and somebody is hosting you and you're obnoxious enough to murder that host before you go home. Well, that's exactly what's going to happen with Mr. Hyde here. The lytic portion of the lambda life cycle ends in the death of E. coli. Okay, the lytic portion of the lambda life cycle ends in the death of E. coli. So there's two phases here, Dr. Jekyll, Mr. Hyde, caterpillar, butterfly, that kind of idea. Okay, so folks, to help you understand this, I'm going to show you like a minute 30-second video on lambda. I've already put the link up in the note document so you can watch it again during your private study time. And it will be a video that tries to explain the basic idea of figure 13.12 in your textbook, okay? So let me cue this up and we'll watch this. Okay, folks, here we go.
When phage lambda infects E. coli, either the lytic or the lysogenic cycle may be followed. In both cases, the first step involves the phage attaching to the host cell and injecting its DNA into the host cell. In the lytic cycle, phage nucleic acid is replicated and phage genes are expressed, resulting in production of phage proteins. Mature phage particles assemble and the host cell lyses, releasing the phage particles. In the lysogenic cycle, the phage DNA is not replicated or transcribed. Instead, the phage DNA integrates into the host cell genome. The host cell can then replicate, carrying the integrated phage genome. The integrated DNA is referred to as prophage DNA, and the host cells carrying the prophage DNA are said to be in the lysogenic state. When the cells are exposed to ultraviolet light or to certain chemicals, phage induction occurs. The prophage DNA is excised and the phage enters the lytic cycle.
Okay, folks, so a pretty short, sweet cartoon involving this topic that we're going to spend a few minutes talking about. As I get my workspace organized here again, do you have any questions from the video that you just saw? Okay, all right, so here's what we're going to do. We're going to walk through each of these phases and make some characteristic statements, take some notes about them. Okay, and so let's talk about the lytic cycle. Okay, the lytic cycle of bacteriophage lambda. You can again look at figure 1312 or figure 1311. Figure 1311 is a bit more detailed, and so I'm gonna use some of the terminology on figure 1311 to carry this conversation. All right. So let's talk about the basic steps of the lytic cycle. Remember, with the lytic cycle, okay, with the lytic cycle, the host cell is going to die. So we're talking about Mr. Hyde right now. Okay. All right. So very first thing that's going to have to happen, and it was shown to you in a cartoon version in the video, is that the virus has got to stick to the surface of the host cell. That's called attachment. Okay. So in attachment, the phage attaches to the host cell by binding to a receptor on the surface of the host cell. Okay. I mean, one of the most important things to do whenever you discover a new virus, or there's a new virus like coronavirus that you're interested in, is figuring out, well, what is the receptor on the surface of the host cell that the virus uses to attach? Because if we can identify a drug that interferes with that binding event between the virus particle and the surface receptor of the host cell, maybe we can block infection. I mean, that's why it's so critical to do this. Okay. So what I'll say here is with lambda, the host cell receptor, the E. coli receptor, is the maltose receptor. The E. coli receptor that lambda attaches to is the maltose receptor. Okay. All right. All right. So Taylor, if I could please ask you to unmute your microphone here for a second.
Sure.
How you doing today?
I'm doing all right.
Okay, Taylor. So we're saying here in our conversation with lambda that lambda attaches to the maltose receptor on the surface of the E. coli cell. So Taylor, my first question to you is this: what is the purpose of the maltose receptor?
I'm not sure.
Okay, Taylor, stay with me. I'm going to lead you to the answer, okay?
Okay.
So Taylor, here's my question to you, and this might seem silly, but it's philosophical.
Okay.
Taylor, who is buried in Grant's tomb?
Grant.
Okay, Grant is buried in Grant's tomb. So I'm suggesting to you that the answer to my question is right in the question. What is the purpose of the maltose receptor?
Is it to receive maltose?
Yes, it is, Taylor. Very
All
good.
right.
Very good. Stay with me. Okay. So Taylor, what is maltose? Can you tell me?
Is it a sugar?
It is a sugar. That's what I'm after. A maltose is a carbohydrate. So define maltose, a type of carbohydrate. Okay. So okay. So Taylor, E. coli has this receptor, which we call the maltose receptor. It is expressing this receptor. In other words, there's a maltose gene in the E. coli chromosome, which tells E. coli cells how to manufacture the maltose receptor that ends up sticking outside the plasma membrane of the cell. So Taylor, my next question to you is this: is E. coli purposefully manufacturing the maltose receptor to enable itself to be infected with lambda?
I'm sorry. What was the question again?
Sure. Let me type it out here and I'll say it at the same time.
Sure.
Is E. coli intentionally manufacturing the maltose receptor in order for lambda virus to infect it?
I would say yes, it is.
Okay, thanks, Taylor. I appreciate it. And I got you. I led you down a path that led to the wrong response from you, but I do appreciate your participation today. What I'm trying to get everybody to see here is that the purpose of the maltose receptor is to allow E. coli to bind maltose sugar in the environment, internalize the maltose sugar, and break it down for energy. That's the whole purpose of the maltose receptor. The purpose of the maltose receptor from E. coli's perspective is most definitely not to enable some kind of questionable beast out in the environment like lambda virus to get inside. That's not why E. coli is doing this. Okay, let me type this out. So first off, let me expand upon the first question. What is the purpose of the maltose receptor? To bind maltose, a type of carbohydrate, so that maltose can be metabolized by E. coli to make ATP, to make energy, okay? So my next question is this: is E. coli intentionally manufacturing the maltose receptor in order for lambda virus to infect it? Okay, no, it is not doing that. Okay, it's not doing that. Let me state this a different way and I'll be ridiculous. Do you have a front door at your home so that a burglar has an easier time getting into your house? Right? That's silly, right? That's silly. Now, some of you might have said yes, because you know you have a weird sense of humor. But no, that's not the reason. So Kit has a question there in the chat window. Kit's question is, did the virus just figure out how to take advantage of the existing receptor? Yes, Kit, it did. In fact, that's how viruses work. Viruses take advantage of other molecules that the cell is using for other purposes to gain access into the host cell. And that's the point I am trying to get here with this conversation. Okay, so from E. coli's perspective, the maltose receptor allows it to acquire maltose. E. coli is not interested in facilitating its own infection with lambda. Okay, so number 4 here, or now, no, just another bullet point here. Number 4, okay, lambda, as Kit suggested in the chat window, is taking advantage of the presence of the maltose receptor to have something to grab onto so it can gain access into the cell. Lambda has evolved a way to attach to maltose receptors to gain access to the cell. The cell did not evolve. The cell did not evolve maltose receptors to enable its own infection. It just so happens that a virus evolved to take advantage of the fact that there is a maltose receptor. Okay. Okay, folks, do you have any questions so far? If I could type better, that would look normal. Okay, all right, let's continue here. All right, so that's the first thing. Lambda attaches, and it does so by using the maltose receptor. Okay, next, part B. Next thing that has to happen, and again, use figure 13.11 for visualization. Next, we have penetration. Okay, take a look at that. The virus has to inject its DNA into the host cell. Now, that's not a trivial thing to do. I mean, it looks like it's a simple thing to do in a cartoon video that I showed you, but it is not a simple thing to do. So in penetration, lambda injects its own DNA into the host cell. Okay. So obviously for lambda to get its DNA into the cytoplasm, it has to be able to punch through not only the plasma membrane, but a cell wall, right? Because E. coli is a bacterium and it has a cell wall. So what I'm about to tell you is that before lambda can inject its DNA into the host cell, it actually releases an enzyme called lysozyme. It releases an enzyme called lysozyme that floats down onto the surface of the cell wall of the bacterium, and the lysozyme begins to enzymatically break down, begins to enzymatically degrade, it begins to chew on the cell wall like a Pac-Man and chemically disrupt the peptidoglycan layer. Okay, before lambda can inject its DNA, the virus releases an enzyme called lysozyme onto the surface of the cell wall to enzymatically break down the cell wall. It's kind of like if I'd use a boxing analogy, it's kind of like having a couple body blows to the abdomen to weaken your opponent, right? You need to weaken the opponent. You need to soften the cell wall so the virus can actually inject its DNA. How do you do that? You release an enzyme called lysozyme, which breaks down that cell wall and softens it up. Oh, that's horrible. Okay, let's see here. I need to ask some more questions. Okay, let's see here. Arlen, could I chat with you for a second?
Hello.
Hi, Arlen. You doing okay?
Yeah, I'm okay. My allergies are just really bad.
Okay. So Arlen, let me ask you, when a lambda virus particle is sitting down on the surface of the cell wall of an E. coli cell, where did that virus particle acquire its lysozyme?
I'm not sure.
Okay, so there's a bit of philosophical discussion here. So, Ireland, do you share any features of your body or personality that when you're around people say, oh, you remind me of your mom or dad?
Yes.
Okay, so can you answer my question now?
Would it
Go
be
ahead.
from the lambda? Sorry.
Okay. Thanks for participating, Arlen. I don't want to make you cough anymore, so thank you. Let me type this out, and that was not a bad response. So let's think about this. Okay, when a virus particle is attached to the host cell wall, where did that virus acquire the lysozyme? Well, there's a couple things I want you to appreciate. First off, that lysozyme was already packaged in that virus's capsid. The lysozyme is present in the viral capsid. How did it get there? Okay, so this gets to my philosophical question, Arlen. The virus that we're currently talking about, the lysozyme that's in its capsid, was put there when that virus had been newly manufactured in a previous host cell that had been infected. In other words, everything in the virus that is present in the structure of the virus that's sitting on that host cell comes from having that virus been manufactured in a previous cell that had been previously infected. Okay. Folks, question in the chat window. Am I making sense? Does it make sense that the virus sitting on the host cell acquired its molecules from a previous cell that was infected with the virus? Okay. Let's see, I need to put that into your notes. How did it get there? Okay. The virus in question has its molecules placed into it as the virus was newly made inside a previous host cell that was infected with the mother of this virus. Okay, so a previous host cell had been infected with an ancestor, mom and dad viruses, not literally mom and dad because there's no gender here, but I'm using it as an analogy. The virus in question that's sitting on the host cell, it came because it was manufactured when this virus's mother infected a previous host cell and caused the previous host cell to make all the building blocks necessary so that the virus we currently are talking about could be manufactured. Okay, I'm just reading my notes. That seems okay there. All right. So another question. Let's see. Geneva Potter. Geneva, can you say hi to us, please?
Hi,
Hi, Geneva. How are you today?
I'm okay.
Geneva, here's my question for you. The lysozyme enzyme that I'm talking about in our discussion here, for the cell to know how to make that enzyme, what must be present in the viral DNA?
Wait, can you repeat the question? You said in order for the
Sure.
enzyme. Go ahead.
I'm gonna restate it for you, Geneva, and I'm also typing it at the same time.
Okay.
For a host cell to understand how to make lysozyme, what must be present in the viral DNA?
I'm not sure.
Okay, Geneva, I appreciate you trying though. I really do. Thank you for trying today, okay? So,
No problem.
okay, folks, let's open it into the chat window. If you think you know the answer to this question, please type it into the chat window. For a host cell to understand how to make lysozyme, what must be present in the viral DNA? Okay, all right, thanks for those that are contributing. So far, take a look at Chelsea's comment. Chelsea's comment is this: specific nucleic acid code that codes for the protein. Chelsea, please unmute your microphone.
Hi.
Hi, Chelsea. Hey, Chelsea, what do we call a specific nucleic acid code that codes for the protein?
Oh geez.
No, that's
I
not
was
it.
thinking, I don't know, I was thinking coding segment, but that's not right.
No, you're overthinking it.
An amino acid? I have no idea.
Oh, come on now. You're doing so good. All right, let me give you a hint, Chelsea. Starts with the letter G.
A gene.
Yes, there
There we
it is. Thank you, Chelsea. Okay, good. All right, good. All right, so folks, when you're taking a class, 9 times out of 10, the reason you cannot answer a question correctly is because you have the deer in the headlight moment because your teacher is actually asking you something. I firmly believe most of you know these things, but we need to pull it out of you, right? We need to draw it out of you. Okay, so good job. So for a host cell to understand how to make lysozyme, what must be present in the viral DNA? The viral DNA has got to have a lysozyme gene. It's got to have a gene. Okay, and I think when you see that, you think, oh, yes, that has to be the right answer, okay? All right, any thoughts or questions? Okay, I'm gonna start moving faster, folks, because I wanted to cover HIV today as well. We'll see how we do here. Okay. Okay. Letter B, penetration, lambda injects its own DNA into the host cell, right? Truth be told, it's not just DNA that goes into the host cell, it's other things as well, like enzymes and things, but for right now, we're just going to keep it simple. Okay, letter C. Letter C is biosynthesis. And here I'm going to wave my hand a little bit and say some things happen. The reason I'm waving my hand and saying some things happen is because the things that happen are incredibly, incredibly complicated. Not that it's not understandable, but it's not quite the level, it's not quite what we want to throw at an undergraduate course yet. Let me put it this way. When I was studying lambda in graduate school, I had a full month of lectures on lambda just by itself. And so I'm giving you a half an hour here, okay? So biosynthesis, long story short, of the molecules that are inserted into the host cell, the molecules turn the expression of E. coli genes off and turn on the expression of viral genes. In other words, when lambda injects its DNA into the host cell, it causes the machinery of the host cell to switch gears, and rather than making the gene products of E. coli genes, the machinery of the host cell starts making the viral products of the viral genes. Okay, so the presence of viral molecules kind of takes over the host cell, forcing the machinery of the host cell to make new viral building blocks, okay? New viral molecules that are needed to build new virus particles. Okay, so that's biosynthesis. And that's actually something we really do understand. But as I said, it's not quite what we usually throw at undergraduates. All right. So the presence of these viral molecules takes over the host cell, forces the machinery of the host cell to make new viral building blocks. Okay. Letter D, maturation. Okay, maturation. The building blocks that have been manufactured. Okay. Actually, before I go any further, I'm going to ask a question here. Right, I know it's going to like, damn, it's another question. Okay. All right. So let's have Afala Shadi. Afala Shadi, could I say hi to you, please?
Hi,
Hi, Afala Shadi. How are you today?
I'm good.
Afala Shadi, let me ask you a question. For letter C, biosynthesis, when the virus needs one of its proteins manufactured, Afala Shadi, is the virus going to use its own ribosomes, or is it going to use the ribosomes of the host cell?
I think it's going to use the host cell.
You think it's going to use the host cell, Afalashadi?
Yes.
Okay, ding, 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 you won the prize. You're right, Afalashadi, it's going to use the host cell ribosomes. Thank you for participating. It's going to do that because, right, viruses do not have their own ribosomes. Okay. All right. So again, remember, viruses basically take over the machinery of the host cell and get the machinery of the host cell to do its bidding. Letter D, maturation. All the building blocks that are synthesized during the biosynthesis phase of the lambda infection will be assembled and put together into new virus particles. Viral subunits are linked together. And when we say linked together, you should be thinking about the last time that you played with Lego bricks and you put bricks on top of each other to build a Lego robot or a Lego dinosaur or a Lego dollhouse, whatever it is you like. The viral subunits, the molecules come together, they are assembled into new virus particles. Now, as animals, when we're born, right, we're infants, and then we go to toddler, and then we go to, childhood, then adolescence, then finally we reach adulthood, right? There's a developmental phase for us as human beings. There is no developmental phase for viruses. They are created, they are born as a mature, complete adult virus particle. There is no maturation. There really is no developmental process. You build them, you put them together, boom, they're completely, fully competent virus particles. Okay. So the viral subunits are linked together to build complete new virus particles capable of infecting new cells. But obviously at this point, that new virus particle that's inside the cell that was just made, it's inside a cell and it wants to get outside so it can infect other cells. Well, how does it do it? It's got to get back through the cell wall. You guessed it. How's it going to do it? Some of the, Tiffany, thank you. Take care of yourself. Let me know if you need anything. How does the virus do this? It's going to rely on additional lysozyme enzyme that's been manufactured in the host cell to nibble on the inner surface of the cell wall, soften it up, degrade it, break it down so the virus can push its way through the cell wall and be ejected and released from the host cell. That is letter E, or release. Okay. New viral particles are released from the host cell when lysozyme softens the inner surface of the cell wall. Okay, folks, that in a nutshell is the lytic phase of lambda infection. And what we should say here in big bullet points is when the new virus particles leave the host cell, they got to damage the cell wall to do it. When the cell wall is damaged badly enough that new virus particles can get out, the host cell dies. It does not recover from that event. Okay. The damage to the host cell wall is irreparable, and the host cell dies. Okay, the host cell dies. All right. Before I go any further, do you have any questions about the lytic cycle? Okay. So, even though our own human cells don't have to worry about this happening to our own human cells, E. coli, including E. coli inside our body, does have to be concerned with this. Okay. Let me ask you a question in the chat window. Here we go. Do you think bacteriophage viruses that kill bacterial host cells could be used as medicinal therapy to treat human infections caused by pathogenic bacteria? All right, good. What I'm trying to get at, and I think what you're thinking about, is that yes, if this virus kills the bacterium and there's a bacterium that's making the human sick, if I put the virus into that cell and kill that bacterial cell that's making us sick, maybe that would cure our disease. Maybe that would cure our infection. In fact, folks, that has been done for a very long time in various parts of the world, particularly Russia and in former nations of the former Soviet Union. No matter what you think of international relations between countries, Russia has a very superb expertise when it comes to virology. And I can tell you for a very long time, Russian scientists and doctors have been treating bacterial infections in humans with bacteriophage particles. It hasn't caught on so much in Western medicine because Western medicine is corrupt with capitalism and things like this, where we want to reap as much profit as possible out of some things. And I'm not joking about that comment. Truth of the matter is, there are other ways that you can deal with infection, and for better or worse, we choose not to do that in our capitalistic Western society. But there have been documented cases of untreatable infections in the West of patients taking a trip to, Russia and other places in the East, and actually having their life saved because scientists were using bacteriophage bacteriophages to treat the infection. So it's something that maybe we'll see at some moment in Western medicine, but it could be a long time because of some of the corruption in our system. Okay. So Aubriana, you say in the chat window, so would this be better than antibiotics, and would it help stop antibiotic resistance? Aubriana, it would certainly be beneficial to slowing down the development of antibiotic resistance, without a doubt. That it would be beneficial for that. Would it be better than antibiotics? Well, define what better is. Better, in some cases it would be more effective because you might have a species of bacterium that is resistant to every known antibiotic, and so there's no way to cure the patient. But with the right bacteriophage, we might be able to cure that patient. So it's better, but it's not perfect. Okay. All right. Let's continue here. We definitely got to wrap this topic up. So let me go to the lytic cycle. Okay, so this will be number 4. I'm sorry, we did the lytic. Let me go to lysogenic, number 4. Lysogenic cycle of lambda. And for that, really the only thing you can look at in your book is figure 1312, but that will suffice. That will suffice. And I'm just going to give you a couple talking points about the lysogenic cycle. The lysogenic cycle, what should I tell you? Okay, for the lysogenic cycle, okay, I want to break it down into stages again. So letter A is attachment. And with the attachment phase of the lysogenic cycle, the exact same series of events that took place in the lytic cycle occur. So attachment is the same as the lytic cycle. It's the same as the lytic cycle. Next, we have penetration. That is also the same as the lytic cycle. Letter C. Okay. So you'll notice that letter C in the lytic cycle was biosynthesis. But in the lysogenic cycle, there is no biosynthesis. So letter C is not called biosynthesis. Letter C is called insertion. Insertion means this. The viral DNA is inserted into the bacterial chromosomal DNA. The viral DNA is inserted into the bacterial chromosomal DNA. Okay, and so this is where we got to take on a special word here. And you probably picked this up from the cartoon. And I'm trying to see if your book also shows you that. Yes, okay, yeah. In figure 1312, if you have figure 1312 accessible on your computer or in your book, figure 1312, take a look at the text of information associated with green ball number 5. Green ball number 5 or green ball 3B. Let me read the text of 3B. 3B says the phage DNA integrates within the bacterial chromosome by recombination, becoming a prophage. That's the word I'm looking at here. Okay. When the viral DNA is a piece of the host DNA, it is referred to as a prophage. It is referred to as a prophage. Okay. What is interesting about prophage DNA is that prophage DNA, when the virus inserts into the chromosome, it becomes a prophage. And you might think, well, then the virus is going to stay in the DNA forever. That's not necessarily the case. There are circumstances caused by environmental triggers, environmental stimuli, where the prophage can pop itself back out. So it's kind of like having a card that you can put into another deck of cards, but then you can take that same card back out of the deck of cards, okay? So prophage DNA can both become part of the chromosome and it can leave the chromosome. So prophages do have the ability, when triggered by the correct environmental stimulus, to leave the host chromosome. Okay, we'll talk a little bit more about that in just a second. All right, so letter D here. So we've inserted the viral DNA into the bacterial chromosomal DNA. Letter D, DNA is replication of the viral DNA. Folks, the viral DNA is going to be replicated anytime the host cell itself reproduces. So what I'm saying is the viral DNA will be replicated by host cell machinery every time the host cell performs bacterial reproduction known as binary fission. The DNA is copied whenever binary fission of the host cell takes place. Okay, and so that is basically the lysogenic cycle of lambda. I mean, those are the basic steps. So if you're okay with understanding some of the technical details of lytic and lysogenic, there's a very important question that we need to address, and it's this. Okay, what determines whether lambda goes through its lytic or lysogenic life cycles? Okay, what's the determining factor, right? With Dr. Jekyll and Mr. Hyde every time Dr. Jekyll drank some kind of chemical concoction, he turned into Mr. Hyde, right? So that's not what's happening here, but it's an analogous question. Let me put this on a different page here. What determines whether lambda goes through its lytic or lysogenic life cycles? And so, does anybody happen to know? Have you come across any reading of this in your textbook that explains this? Okay, well, just for the sake of time, I'll move us forward on this. Gang, the critical issue is this. What determines whether lambda goes in lytic or lysogenic directly correlates to the host cell and the amount of stress the host cell is experiencing. Okay. The amount of stress the host cell is experiencing correlates to the decision of whether lambda proceeds with its lytic or lysogenic cell cycle. So you can think of it this way. When the E. coli host cell is not stressed, when E. coli is not stressed, then lambda will enter the lysogenic cycle, pop the DNA right into the host chromosome, and just enjoy replicating itself every time the bacterial cell replicates itself. Okay. Whenever E. coli is not stressed, lambda enters a lysogenic cycle. Okay. Whenever E. coli is stressed, lambda can detect that. And you think about it. If E. coli is stressed and lambda can detect that, then lambda is thinking. And obviously this is not a thinking thing. Lambda does not have a consciousness. This is all biochemistry and biophysics here. But lambda can detect the biochemical stress of the host cell, and that causes lambda to enter the lytic cycle, because it seems to be the reasoning of Mother Nature that if the host cell is stressed, then the host cell might subsequently die pretty soon. And if the host cell dies pretty soon, then lambda is going to lose its happy home, isn't it? So the virus takes the approach that if my host cell, which I need to reproduce myself, is about to die, then I better do something to make as many copies of myself as rapidly as I can before the host cell actually dies and falls apart. So whenever E. coli is stressed, lambda will enter the lytic cycle of its life cycle, the lytic phase, and you will produce new virus particles as rapidly as possible. Whenever E. coli is stressed, lambda enters the lytic cycle to make as many copies of itself as it can before the coli dies. Okay. All right. Okay, folks, thoughts or questions about this whole topic? The point we're trying to make here is that lambda has the ability to go through two different processes, lytic and lysogenic. If E. coli finds, if lambda finds itself in an E. coli cell that at that moment is stressed, then lambda will enter the lytic cycle and attempt to make new virus particles. It is possible that when a new virus particle is made and ejected from that host cell, that new virus particle, which is already triggered to go into the lytic cycle when it infects the next cell, will find that when it infects the next cell is actually not stressed. And so when the next cell is not stressed, the new virus particle that thought it was going to go into lytic will switch gears and start going into the lysogenic cycle because the new cell is not stressed. So it goes and incorporates its DNA into the host chromosome of the new cell, and it copies itself many times. The copies stay in the lysogenic cycle until that new cell starts to get stressed again because of a change in the environment, such as a lack of water or exposure to sunlight or whatever else might be. That new cell starts to get stressed, and though lambda detects that, prophage pops out of the viral chromosome and gene expression changes trigger the onset of the lytic cycle again. So the virus particles throughout a population of cells can go back and forth between lytic and lysogenic, depending on the environmental stress that the host cell is or is not experiencing. Do you have any questions? Okay, all right, folks, there's lambda for you. Okay, I'm gonna have to wrestle with HIV somewhat next class period, but we'll be okay. We'll be okay. If you have no questions, all right, do you have any questions about lambda? Okay. Because I do have questions about lambda for you, and they're waiting on the exam. Okay. All right. Let's continue here. We're going to talk about HIV next. Okay. All right. So HIV's life cycle is actually not really presented in chapter 13 here of your book because there are so many viruses. But let me say this. HIV is a virus that's known as a retrovirus. Okay. HIV is a retrovirus. And so the most important figure in your textbook for this at the moment is figure 1319, right? Figure 13.19 is multiplication and inheritance process of the retroviridae. HIV is a retrovirus. I'll tell you what that means in just a moment. Okay. All right, so let me give you some background information here. We're going to be talking about HIV today and obviously on Thursday, and I will also talk to you about the subsequent development of an HIV infection into proper AIDS, AIDS, immunodeficiency virus, acquired immunodeficiency syndrome. Okay, number one. Okay, HIV is a retrovirus. Hey, what the heck does that mean? Okay, here's what it means. Okay, the fact that HIV is a retrovirus means the following. HIV's genome is encoded as RNA, not DNA. HIV's genome is encoded as RNA, not DNA. And so if we remember the central dogma, okay, this is from chapter 8. There's a reason that chapter 13 is discussed after chapter 8. Here it is. You remember the central dogma of chapter 8 was this statement, where you take DNA, make it into RNA, make it into protein, right? That's a simplistic view of things, okay? Well, for HIV, okay, for HIV to copy itself, HIV has to take its RNA and reverse transcribe it back to DNA as part of its life cycle. During the HIV life cycle, HIV genomic RNA is reverse transcribed into DNA. In other words, this flow chart here shows you DNA to RNA, but I'm telling you that for HIV, it's going to be RNA to DNA. Okay. So it would be kind of like this. Okay, RNA goes to DNA. Notice the direction of the arrowhead. Okay. All right, that's the first thing that you must appreciate. Okay, I guess I'm just about out of time, but so what we will start with next time is I will describe some of the basic physical characteristics of the virus particle known as HIV itself. I'll show you a video about the infection process. We'll walk through it together in your notes, and we'll talk about how HIV infection leads to AIDS. We can definitely finish that next class period. We can still be ready for our lecture exam a week from this Thursday. Gang, do you have any questions right now? Okay. Hey, thanks for your time and attention. I really appreciate it. Look for an email for me later this afternoon. I will include your study guide, okay? Take care, folks.
Thank
Thanks,
you very
Andrea.
much.
Thank you.
Welcome. Bye-bye.
Thank you.